Parkinson's disease is a neurodegeneration which is supposed to begin by cell loss, neuronal loss in the dopaminergic cells of the brain.
The patients with Parkinson's disease, in addition of having dopaminergic cell loss, what they have in the remaining neurons of their brain is an accumulation of different proteins inside the neurons that are called Lewy bodies and are really the signature of the disease.
For being classified as suffering from Parkinson's disease, you should, 1, exhibit the Parkinson symptoms, so the slowness of movement, the inability to move, and some patients might also display the tremor. But after you died, when the pathologists look at your brain, you should have the presence of Lewy bodies all over your brain.
In the 1990s, it was found that mutations in the gene of alpha-synuclein could cause a familial form of Parkinson's disease.
Not only synuclein was causative of the disease, but it soon appeared that synuclein was a major constituent of the Lewy bodies, which are the pathological landmark of the disease.
Because aggregated alpha-synuclein from Lewy bodies are the signature of the disease, we wanted to know whether this pathological alpha-synuclein in Lewy bodies could have the potential to initiate or extend the disease.
How we demonstrate this? And we demonstrated this by first purifying synuclein obtained from the brain of PD patients. So we were sure that this synuclein was somewhat related to the disease itself, as we were taking it from the brain of PD patient. And then we injected this purified synuclein to cell culture, to rodents, or to primates.
What we have found also, it was that the human alpha-synuclein that we injected into the animals was able to transform the host alpha-synuclein from the animal from a normal state to a pathological state. And this pathological alpha-synuclein, this now becoming pathological alpha-synuclein from the animal, adopted an aggregated conformation and accumulated within dopaminergic neurons, and also was able to transmit this pathological capacity to other cells and in other brain regions.
So we had in hands the first demonstration that indeed the misfolding of the synuclein, directly taken from the brain of PD patient, was capable of causing the death of dopamine neurons. While we had demonstrated the proof of concept, we wanted to enlarge, to increase the statistical power of our demonstration, and therefore we needed to work on a much larger number of animals.
We have the opportunity to work with the baboons colony in Murcia, so with 50 monkeys, so it's a huge quantity of animals, and with this group we can decide to make a different experiment with the same colony. The big challenge was to move other surgery materials for the 50 monkeys, because 50 monkeys to operate is quite challenging, and I think it's the only way and the first time that a group of research in the world has done that.
What we are testing is we are injecting these Lewy bodies not only in one region of the brain, but also in different regions to be able to dissect how the pathways of transmission of the alpha-synuclein pathology may work, and not also inside the brain, but also from the periphery. So we are injecting also the Lewy bodies in the duodenum and the stomach, and in order to determine whether this alpha-synuclein injected in the stomach or the duodenum may be able to reach the brain.
What is totally new was to deliver the Lewy body material into the gut of the normal animals. This is important because one hypothesis stance that the very beginning of Parkinson's disease occurs at the peripheral nervous system, particularly in the gut, and the toxic protein travels all the way up to the brain where it causes the damage. If we could reproduce such an effect in our monkey models, not only we could have a very model of humans' Parkinson's disease, which could prove that the theory is correct, but particularly it could give us ample room, ample possibility of stopping such a process.
We'll see in 2 years if there is success of propagation, if we can see Parkinson in monkeys after gut administration.
We believe that this project may provide very important clues about the initiation of the disease and also about the extension and the progression of Parkinson's disease, which is currently unknown.
If we stop the cell damage of the substantia nigra well before it can occur, this is close to almost saying we can cure Parkinson's disease.
Such a demonstration will definitely be a revolution.